I'm FLT3. Look, I'm mutated! I'm gonna kill you. Attack!
What is FLT3? FLT3 is a kind of receptor tyrosine kinase. It is encoded by a gene called FMS-related tyrosine kinase 3, or FLT3 gene. FLT3 protein activates pathways in cells that are important in cellular proliferation. FLT3 is a type 3 protein, which contains two distinct bands corresponding to 1300 and 1600 kDa. The larger band represents the functionally mature fully glycosylated form. It has an extracellular ligand binding region with 5 immunoglobulin-like domains and a single transmembrane domain. The cytoplasmic portion is comprised of a juxtamembrane domain or JM domain, followed by the catalytic domain, which contains activation loop and ATP binding pocket. The catalytic domain is interrupted by a kinase insert domain or KID. The presence of the complete JM domain helps FLT3 to adopt its autoinhibited conformation. For its crystal structure, FLT3 is like other protein kinases. It consists of N and C terminal domains or N and C lobes. The N lobe contains a twisted 5-stranded anti-parallel beta sheet adjacent to an alpha helix. The activation loop is attached to the C lobe and contains 2 additional beta strands with a small beta sheet that is characteristic of inactive kinases. Several amino acids play important roles in stabilizing the conformation of FLT3 in the autoinhibited state or the normal state. Aspartate A39 or D839 and D835 maps to the activation loop of FLT3 and stabilize the conformation by forming several hydrogen bonds, as well as hydrophobic interactions within this region. Isoleucine A67 is located directly at the beginning of an alpha helix and is part of a hydrophobic interaction site that stabilizes the helical bundle, which forms the C lobe of the FLT3 kinase domain. FLT3 is frequently mutated in acute myeloid leukemia, or AML, myelodysplastic syndromes, or MDS, and other diseases. Most FLT3 mutations are internal tandem duplications, or ITD. ITD occurs when sequences of less than 10 to several hundred bases in length are repeated within the JM domain. All duplications are infrared with the number of nucleotides added as a multiple of 3 and always maintain a head-to-tail orientation. Corresponding new amino acids are inserted in the protein, but the coding frame of the protein remains unchanged. The altered JM domain allows the FLT3 receptor to activate signaling pathways without binding of FL, or the ligand. The receptor is always turned on and is said to be constitutively activated. Point mutations have also been observed in tyrosine kinase domain, or TKD, of the FLT3. Almost all of these mutations involve an aspartate to tyrosine, or D2Y substitution at codon A35, although other substitutions have also been identified. These mutations cause a conformational change of the molecule and disrupt its autoinhibitory function, thereby rendering the receptor constitutively active. On the molecular level, FLT3-D839G or aspartate change to glycine at A39 and FLT3-D835Y presumably lead to a more flexible activation loop and consequently higher propensity for tyrosine phosphorylation. The corresponding amino acid D820 of the homologous CKIT receptor has also been described to be mutated in mast cell-related diseases. The mutated form of I867, called I867S, has a serine at this position likely destabilizes the surface region of the helical bundle. I-867S presumably alters the fold or stability around the interaction site to the JM domain and leads to a reduced affinity between JM domain and CELO. As a consequence, this amino acid could reduce the autoinhibition of FLT3 and lead to an increased activation. Please note that there is no available mutated conformation, and this one is made from the normal one. A lot of victims at the first stage will cough and feel dizzy if they got the mutated FLT3. Receptor tyrosine kinase cell signaling pathway regulates a variety of cellular processes, including cell growth, proliferation, differentiation, survival, gene transcription, metabolic regulation, and others. Receptor tyrosine kinase signaling pathway is activated by the binding of a ligand, such as a growth factor to the receptor tyrosine kinase. The ligand is called FL. FL binding induces FLT3 conformational changes. Typically, one FL will bind to two FLT3 monomers and thus form a ternary complex with dimerization of FLT3. FL activates FLT3 by cross-phosphorylation of intracytoplasmic tyrosine residues within kinase domains. Once the receptor is turned on, numerous downstream pathways are activated, including MLP kinase signaling. There are several potential strategies to disrupt a signaling pathway that is always on. The first focuses on neutralizing the ligand before they can associate with their cognate receptors and initiate the signaling cascade, as shown in the video. The second approach is to directly inhibit the receptors, often through the physical blockade of the receptors with directed monoclonal antibodies or disruption of the enzymatic by small molecule inhibitors. The third approach relates to the inhibition of signaling of cytoplasmic second messengers. As we can see, the binding of the inhibitors changes the conformation of FLT3, so it cannot bind to the FL. The initial attempt to treat with FLT3 utilized the phage display library to generate monoclonal antibodies. However, recent development of FLT3 inhibitors has focused mainly on ATP-directed small molecules. The refusion of the initial assumption allows us to discover more efficient drug towards mutated FLT3 with higher selectivation. For future development, the identification of novel predictive biomarkers will be helpful. Second, it will be critical to understand how tyrosine kinase inhibitors interact with the actual leukemia stem cells and whether such cells can be susceptible to biological effects of FLT3 inhibitors. You wanna kill me? Then come.